think I have any specific announcements today. Just remember we're putting the book down for next week, so we're gonna have different reading assignments and more work for the LOs next week. I think I posted the reading assignments for Monday. Still checking into changing a couple of things for Wednesday, so I'll update you with Wednesday's reading assignment. Okay, I created a lot of questions today. So I was like, oh my god, it's like 20 questions in here. But so what I'm going to do starting today is there's two of you that are just one question away from being exempt from midterm 3. So once those people reach their max, you won't be able to answer anymore, but I want to encourage either group discussions if that's possible, right? I don't mind so much that you share what you know, but more than, you share what you know and you can put it in your own mouth and say it out loud. The idea is that more than just reading from the book, sometimes just saying it in your own words helps you clarify the concept. So more than competing and doing it like who raises their hand quicker, I want to give more like overall everyone the opportunity to answer questions. Okay.
Okay, I don't know if this is right, but I have just like, if there's excess light, they're going to move away from it. Like if there's too much, the chloroplasts will move away. But then if there's limited light, they'll move towards the light.
Yeah, but these are metabolic.
So
more than the plant itself. Like again, think about a cell, the plant cell itself. So you're right on that, but weren't there yet.
If there's too much light, photosystem 1 and 2 will shut down.
Yes.
And then
yeah, so that's one
yeah,
thing. Okay. So what about when there's very little light? What could happen? Could be anything of the things we've talked about.
Well, I know what the plant will do, but I don't like as in terms of growing wise. Are you asking that? So if there's little light, the plant will focus more on growing like length maybe and less on leaves. Like it won't grow. The leaves will be smaller because you're not really focused on gathering the light to conduct photosynthesis. You're more focused on growing maybe to reach that light and lengthwise.
So we haven't really discussed too much about very little light, but one of the things is, we can start putting together things as in too much light eventually can shut down both of the systems, because of photoinhibition. And very little light, many things can happen. And again, we haven't talked about it in detail, but let's say if a plant is not efficiently doing photosynthesis, remember that reduced redoxin is also required for nitrogen fixation. So there are just multiple things that a plant is going to have to overcome if photosynthesis is not as efficient. Okay? So we talked about excessive light, right? Just this photoinhibition. And specifically, one of the things that Isabella mentioned is there's a very important part of photosystem 2 that is the one that takes the most light, that does the heavy oxidation process. And that's called B1, it doesn't matter, but once you have that very important part of photosystem to shut down, the whole thing will shut down. So that's photoinhibition. Clearly, this is not 5B. We said, oh, I know why. Because this is from the previous um, lecture. So what happens when plants do not receive enough light? So photosynthesis is inhibited, which in turn affects carbon and nitrogen fixation. It induces chlorosis, slower growth, and phenotypes. And that is basically the last slide we talked about. So we started discussing that last class. So leaf anatomy. We're not talking about the plant, the whole plant. Leaf anatomy. So if you focus on the leaf, how has leaf anatomy evolved to maximize light absorption? Yes.
So chloroplasts are kind of localized in both areas, so they're kind of not balanced, like evenly distributed. Some light can actually fall in between those spaces. Thousand cells can actually reflect light down into the cell, into this kind of mesophyll, and then the irregularities of the air spaces in the water inside there can refract the light to extend the wavelength inside, giving it more picture.
Yeah, so it's a lot of how the whole leaf is organized. So we have, just to review this part, we have epidermal cells, we have palisade cells, and then the mesophyll cells. So one of the things is we're talking about light. And we know that all of these part of photosystems, we've talked about how the mesophyll are like the ones that are doing that most. And then when you think about it, if your cuticle is too thick and it prevents light from passing through the mesophyll cells, then who's going to be doing the photosynthesis? And even when these are microscopic organelles and clearly microscopic molecules, they will generate shade. If we're talking about light, and you are lying on top of a chloroplast, and it is basically shading what's underneath, then if a chloroplast is placed underneath, there's not going to be any light capture from that chloroplast placed underneath. So one of the things is epidermal cells are transparent. Okay? When you look at epidermal peels, they are just translucent. You will see them. There are many chloroplasts there, and they are also convex. So basically, they just help light flow. And then another two aspects that Ethan talked about. Was that just basically the localization of those palisade cells? So they create a sieve effect that passes through layers, right? So you have light. These cells are convex, and then it just allows light to pass until basically something absorbs that light. And also the air-water interface, pretty much, of the vacuoles in the palisade cells and the mesophyll cells are designed to scatter light. So it's like, think of it as putting a bunch of, I don't want to say mirrors, but things that refract light. So it's like light is here, let's start refracting it everywhere, so something catches it, so that we can take advantage of all of the light that is hitting us. Yes, sir.
So obviously the convex epidermis, but they also have the plasticity and the movement fleet. So the plasticity is like the response to light and solar tracking. So leaves can move to be in the sun, or they can also change so they are out of the sun by dipping down or moving out of the direct sunlight.
Yeah, so plants can do a bunch of things, a variety of things. They can do, one of the things is and it's more obvious when we look at a tree, but not so obvious when we just initially think about it. But the branches of a tree are three branches, just branches. And usually the branches at the bottom are, more expanded than the ones at the top. And that's just maximizing the light. If all of the branches were the same size and everybody is like one below the other, basically the leaves that are at the bottom will get some light very early and very late in the day, but not so much. during the rest of the day. So you have the tree branching. Also, some plants, and this is just specific adaptations in some plants, have the ability to survive, to maximize photosynthesis in those very brief periods of the day when they are exposed to sunflex, and those sunflex would be, the light that is just allowed to pass through like a very dense forest. And another thing that we'll mention that is solar trap. So plants can move the leaves through the day in response to if the light is basically coming straight from the top, or if it's coming in a specific angle, then they can move the leaves and adjust to how much light they can absorb. Yes. Yes.
Would be A, because it's kind of the floor of the movement class movement. You're trying to do another ways to get excess light waves.
So sorry, can you say that again?
A, yeah.
Yeah, but what did you say?
The floor class movement, they're moving out of the way because they don't want to damage themselves, or the pedestrians to damage themselves.
Okay, so they are moving out of the way, and how do you think this is favoring them? You're right. How is this favoring them versus this?
So they're basically just increasing the surface area, I would say. Like I said, just kind of moving out of the way so the sun, they're not getting correct sunlight for all of it.
Yeah, so one of the things is picture A, if you have this, you're maximizing light absorption. You have all of the chloroplasts just exposed there to surface. Here, of course, we're only seeing this, but I think some of the chloroplasts have moved or are actually being under some of the other chloroplasts, so not everybody gets light. But also, picture B. Why do you think B? How can you see B associated with these plants getting too much light? Oh, wow. Yes.
You're folding the leaves like the pieces of there.
Yeah, so you have the leaves folding, right? And so now, clearly once you describe it, of course, is yes, there's less surface exposed, and so there's less surface area exposed to the sun. Yes.
Well, there's the two things that were just shown earlier, chloroplasts have a way to minimize exposure to maximize it, and then also folding leaves to minimize the surface area of the leaf. Then there's the xanthophyll, where they alter the carotenoids so that they can release more light as heat.
Yes, so you are, we've talked about different things, so increasing so that carotenoids can dissipate some of that light, the chloroplast movement. What about entire plant? So how do you think, and this is, I don't think this was specifically in this part of the reading, so it's more, can you, from things that you have seen in just plants that are naturally more exposed to light or warmer temperatures, what do these plants do? We've talked about a couple of them before, but to minimize how much light they absorb.
I know these high heat in some areas with thicker leaves. There's
Yes.
also a waxy layer over it.
Yeah, so they would have waxy cuticles, right? Like if you think of succulent plants, cacti, they are less, they are like pretty much waxy cuticles. And so another thing is, I'm not sure if you are aware of, but it's cryones. And that's something that we'll introduce with this, but it's not just associated only with light. Trichomes are these little hairs that are here, and they minimize light that is absorbed by the plant. Okay, they can refract some of the light. And we're gonna talk about trichomes. There are different features of trichomes. They can be involved in protection against herbivores, and so that's something we're gonna be talking about later on. If you're a tiny herbivore and you have a bunch of hairs in your mouth, I mean, that's going to deter you. Sometimes trichomes can be associated with deployment of toxic secondary metabolites. So, again, but in this case, they can help minimize light absorption. Yes. Oh, sorry, yeah.
I'd say like acclimation is gonna be more like long term. It's like plant will have to change its entire like chemistry or its structure. And then plasticity is more like short term of like adjusting to like sunlight or just changing its shape and
So you say activation is more long-term adaptation and
Yeah.
plasticity.
is more like short term trying to like get more sunlight or moving towards light or like something.
So yes, but not exactly the key difference.
Okay.
So it is talking about, so acclimation is for the most part, it can be short-term and long-term adaptations in that sense. It's not necessarily, keep in mind. It's not necessarily defining if it's short-term or long-term. But they are both associated with adaptation. Can you tell me the difference, Ethan? I think you just raised
I
it.
was gonna say the plastic is like temporary kind of idea of like simplex or just like weak movement and acclimation is just getting used to the environment that they're already in. It's basically like I know the example he has an example of like low temperature and high temperatures. Some plants in low temperatures will do better in low temperature, even though it's the same species. The plant in different temperatures, the higher temperature one will be better at higher temperatures if it's below low temperature, something like that.
Yeah, and so again, that's also partly right. And I think the thing is that these two terms, I sometimes have to admit I use it just interchangeable, like sometimes acclimation, sometimes plasticity, but there's a key difference between both of them. Acclimation is the adaptation itself. Plasticity is how much a plant can adapt. Okay, and that's the key difference. So acclimation is the adjustment. How the leaves can adjust, long term or short term. The entire tree is adjusting, the leaves are adjusting. Plasticity is how much can you adapt. All organisms are capable of acclimation, some extent. How much you acclimate, how much you change is plasticity. Okay? So think about you're growing your plant indoors, and then you transfer it outdoors, assuming you will die. What kind of changes, either cellular level or the overall plant, do you expect to see in that plant that is somewhat under shade or low light, and now it's full-blown light outside? Yes.
The colors of the leaf will change.
So how do you think it will change?
I think they'll change because if it's going from being indoors to outdoors, it's receiving more sunlight. So I'm assuming that there'll be more chlorophyll within the plant.
Okay, so that's a good point. So let's take this leaf that you have here. This is your plant growing there, and these leaves have a certain color. And then you take them outside. How do you think this is going to change now that there's more light? Are they going to be greener or less green? Or how do you think this that we're under sort of light are going to react once we are exposed to light?
Would they be greener
Why do you think they would be green?
because they're getting more sunlight?
Okay, so they are getting more sun, and I'm not saying you're wrong. I mean, maybe plants can vary. So we're thinking about this process, but the way I'm thinking about it is, okay, potentially they could be greener. However, if they are adapted to low light and everything is going full-blown systems under low light, and then you expose it to full light, you might get photoinhibition. Because now this is too much light. Right? It will depend on the plant. Some plants cannot adapt. And so what happens is when you have a plant growing indoors, the full leaf develops to adapt to this light. Everybody's working at good conditions under low light. If now you double the amount of light, then probably that can cause photoinhibition and the leaves can change. And I don't know which way because clearly plants are going to be different, but my assumption is they will go pale because the chlorophyll itself will be killed by the excess of photons outside. Okay? So what plants will need to do in that case would be, for example, these leaves will probably die. And think about plants don't have cell migration. They cannot just, okay, let's replace these leaves or these cells with other cells. The best chances your plant has is Canada, kill these leaves and develop new leaves with better conditions suited for the highlight. So when you do drastic changes to your plant, maybe that's where you see more new growth because that new growth is better adapted to the new conditions. Once the leaf is fully developed, there's very little that the plant can do at that point. It's suited for that specific condition. So usually what happens is when a plant goes from is in little light, it usually develops more chlorophyll because it needs more of those molecules to absorb the light to catch the few little light that is available to them. And they will grow thinner leaves because again, you want to maximize how much light is absorbed. And when you transfer them to high light, something they will do is they will have to develop more quenching pigments, the ones that dissipate in excess light. They will probably develop thicker leaves to adapt to that new light. And also, they will, usually one of the things is they develop more rubisco because now they have more light. And light and rubisco go hand in hand. So if you have more light, you can probably fix also more carbon so you can grow more. And then when they go from high light to low light, they will, some of the leaves will fall and they will develop thinner leaves and will have more chlorophyll. So it's just transitions. Then again, how much a plant can do it. Some plants cannot be changed. They need full light all the time or they will die. But again, the plasticity, how much they can change is going to be in each plant. Yes.
So I remember we talked about like cyclic photosynthesis, and I was in response was it like high light conditions that usually is cyclic photosynthesis?
Yes. So high light conditions can induce the cyclic one to prevent. It's one of the things that a plant can do before killing. So when a plant has fully developed leaves, so the fully developed leaves they cannot change, but also remember about the mobility of nutrients. They are the source in this moment. If you kill all your fully developed leaves, the ones that are developing, that still can change somewhat. Really, they are still sinks. So if you kill all your fully developed tissues, the whole plant is going to die. So yes, so the cyclic photosynthesis is something that can be developed instead of the linear one to prevent so much light absorption.
And then also so they have like the old leaves are gonna be like the sink of, or sorry, the source of those nutrients. Are they like transporting out as many as they can before killing that leaf?
Yeah.
Is that
so
why like they kind of get chlorotic and stuff? Or?
Yeah, so one of the things is when we talk about senescence towards the end of the semester, it's before when a plant starts, when the leaves start senescing, it starts getting pale and then eventually brown. And so what the plant is doing is, with different hormones, is recycling all of this that is still viable, that it can be recycled, putting it back in the phloem, so that other leaves can. So clearly there is certain carbon, certain nitrogen that is just there, that it cannot just disappear. But the plant will recycle all of those nutrients before you kill it. That's something that plants will do, that the trees are doing at this moment.
So it'd be like bluish wavelengths, right? Because if you're exposed to a lot of sun, that's a lot of energy as heat, so you're probably going to be able to produce a quick photosynthesis, and you're going to be absorbing lower wavelengths then.
So one of the things is, similar. So whenever it's hitting the top of the canopy, it's the visible spectrum. Yes, it's all of it. Let's say somewhat similar proportions. So you're right. It's going to be, all full blown blue. But which other besides blue? Which other color do plants absorb?
Red.
They absorb red, right? So clearly, all of this, even UV and infrared, everything is going to be available. But when you think about a leaf at the top of a canopy, then it's going to have everything it wants, so they can choose. It's like, okay, you remember that plants absorb mostly blue wavelengths and red wavelengths. Okay. Yes.
It's going to be your lower wavelengths because it can actually penetrate and go deeper. Whereas in the your higher wavelength ones are going to be all closer to the surface because they can't puncture as deep.
Okay, so which wavelengths do you think are going to be available?
So like your blues would be available.
But if the top ones absorb the blue,
And I guess they could get some from the unabsorbed frequencies. So it would be if the top ones are getting hot enough and they're emitting the red, then red's available as well as probably I guess a little bit of yellow and green.
yeah. So that's so. Of course, we're here without measuring, so we're just doing it empirically and saying, well, if the plants that can pick and have everything available will prefer blue and then red here, then the deeper you go into the canopy, then the less plants don't have so much the option, but what is left. They cannot move, they cannot do much. So what they have is going to be what is left, and for the most part, what is left is going to be somewhere in this region, right? Green and yellow, because they don't really absorb in the green and the yellow. They absorb somewhat in the orange carotenoids and all of these things. Can absorb somewhat here and in this area, but not so much in the green and in the yellow. So now, what about red? What is the maximum wavelength that you remember plants absorb?
It's 700.
It's 700, right? So plants can absorb usually at the maximum at 700. So there's also part here that is still red and moving into the infrared that is not absorbed by plants. And why is that important? Because so we said chlorophyll absorbs strongly, at blue and red. But under high plant densities, plants are going to be exposed to a lot of what is left here at the tail that is called far red light, that is nearing the infrared. And why is that important? Because that is a signal for plants that they are in the shade. Plants can sense if I am getting a lot of far red light and not so much red, so the ratio red to far red is an indication that days are getting shorter or I'm in the shade and I need to do something because, again, either days are getting shorter or I am in the shade, so I move or I do something because we need to do something. Yeah.
How are they sensing those frequencies?
Yes. So I'm gonna tell you that.
Okay.
So this is how plant basically how you can measure the light that reaches plant. And more than remembering all of these is just because the book talks about that. And some of the articles that are assigned to you for next week mention PPFD a lot. So I just wanted to tell you what it is. And it is basically how much light, how many of those, or the proportion of those photons emitted by the sun that can be utilized by the plant. And so we know the plant absorbs mostly 400 to 700 within that range. So how much of that light is hitting plant at a particular time? That's what it is, just basically the light that is absorbed. And then if you were to measure something, the top of the canopy, you can have as much as 2,000, and then here you can have as little as. So even plants will have to adapt to those environments, and these plants that survive in this heavy shaded area still have to be able of adaptation or at least be level down. So, this is Arabidopsis. If you have never seen Arabidopsis, this is the model plant organism. This is grassy, so just different plant, So we have broccoli and cabbage and many things derived from this. So one of these, left or right, they were exposed to higher ratios of red far red, or low ratios of red far red. Lower ratios mean more far red than red. Which plants, the one on the left, or the one on the right, or the one on the left, or on the right, do you think are showing characteristics of a plant that is on the shade, that is sensing shade? And why is it? Yes.
Figure A would be the one on the right because the one on the left hasn't had to grow or adapt and it's getting the proper amount of sunlight, but it realized it had to go up to try to find more, didn't find more there, so it went to the left to try to get out of the shade. And the same is with figure B on the right because it had to grow up and get the extra leaves that went out more to be able to get more sun.
Yes, so basically this is what the plants are trying to do in response to shade. They are elongating internodes, they are trying to search for the light. In this case, they are not really under the shade. What scientists did is they increase the far red, and so the plant is thinking it is in the shade. And the way they feel, so one of the things is, their chlorophylls can still absorb that. It's just not the maximum absorption spectrum, so they can basically, with the same photosystems, they can absorb some of that light. It's just because the energy is little and it's just less usable than the red light. They know that there is shade there. So this one is showing the ones in the right, yes, are showing kind of shade characteristics. Besides the long internodes, can you tell any more differences between the plants on the left and the plants on the right? Yes.
In figure A, represents on the plant on the right, the bottom leaves are a lot thinner and they're stretching out more.
Yes.
And then the same with figure B, where it has like the longer stems and then further spread out leaves than on the left.
Yeah, so the internodes, the whole thing is elongated. The internode so between nodes is elongating. Can you see anything else that is different between both plants? Yes.
Like the colors, I guess.
Okay.
At least in B, you can tell that the one on the right is lighter than the one on the left. And I'm assuming that has to do with the chloroplasts.
Yeah, well, yeah, I mean, I didn't notice that before. This one seems lighter. I can tell you many times yeah, I think this one is definitely lighter, which is maybe just a sign that it's not very healthy. Anything else developmental wise that you see different between them? Yes.
And A, the one on the right has flowers, and the one on the left doesn't.
Yeah, so you can see, at least for some plants, flowering also seems induced by far red light. Why do you think a plant would think, oh, there's a lot of far red light, I better flower now? Yes.
I'm gonna die.
Yes, and so,
It's time to get a seed to not end the lineage.
yes, so that's one of the things that also regulates flowering. I am going to die. The days are getting shorter, or there's shade, or I am not getting what I need. I need to reproduce because I am going to die. And so that red to far red is also important, and the ratio is regulated by plants to control flowering. So, however, flowering, and we're going to talk about flowering. I don't really know if we can talk about flowering, but we can talk about in plant physiology. But one of the things that triggers flowering, well, there's hormones, but there's also photoperiodism, so the length of days and nights. But at the same time, just if that wasn't enough, you've probably seen plants like chrysanthemums that as days get shorter, they flower, right? That's many of these flowers that we have right now in the fall are doing that. But what happens in the spring when days are getting longer, there are plants that also flower. So it's not that simple, but so there's basically different things that regulate it. But so there's a large number of plants that increasing red far red ratio will tell them we're gonna die, it's time to reproduce. Unless you're a long day flower, in which case if you flower at this moment you will die, right? You're not ready to do that. So you will actually have to flower when you have longer days. But all of these to introduce you to something known as shade avoidance response. And everything we've talked about. So the far red light makes plants think that they are not getting enough sunlight. This far red light can also be perceived by the photosystems. It just doesn't excite them as much because it's beyond the wavelength of absorption. And so it induces, in this case, you can see, for example, in this case, if everybody's getting sort of the same amount of light, they will grow the same. But plants like this one in here, because the top ones are getting most of the light, it will make it think it's not getting enough light. So it will induce certain things like elongation, increased leaf size, and apical dominance, which is just grow tall. And grow is taller and get light. And not everybody else is allowed you to get. So the association between carbon fixation and the amount of light can be measured by, light response curves.
So if you're doing it for the different plants, you probably want to standardize like, the light conditions across all four of them. Is that what the
Yeah, so basically, let's say you have 4 plants in your house, and you want to know if they are getting enough light, if they are suited for that amount of light they are getting. How can you, what could you measure? And of course, this is not something you do at home, when you plant your plant, but what are some of the things you can measure? Either describe them or name what the measurements you could take to see if that's enough light for your plants. They are doing fine.
I think you could measure like chlorophyll density.
Okay.
So if they have like extremely dense chlorophylls, then that would probably indicate that they're like kind of on like the border of getting enough light.
Okay.
Versus having too much, they would have like less dense chlorophyll. And then you can measure things like just like quantify like new growth, like kind of growth rates, like whether or not they're budding or like flowering, that kind of thing.
Yes. So one of the things, those are things. So in this case, it's like, what could you do in terms of you? This is not like an organized experiment in which you have 10 plants of the same and you're exposing them to different things. So what kind of things, either visual or measurements, can you take? So you could take probably check chlorophyll. They would help you. But what other things could you measure? Yes.
You could measure like how much oxygen they produce.
Okay.
If you slowly increase the light on them, so they can see if they flatten out basically, to see their maximum photosynthetic rate.
Yeah, so if you want to get more specific and you want to measure actually how much photosynthesis is happening, you can measure output of oxygen, right? Or you can measure basically the CO2 fixation rate. Is CO2 being released? If CO2 is being released, that is telling you the plant is doing more respiration, that's CO2 fixation, right? So in that case, the plant is not going to be happy because yes, the plant needs respiration, but clearly it eats by CO2 fixation. So basically, you can develop these light response curves. So can anyone tell me anything about that respiration rate? Anything you remember from it? Anything that this is telling you on the graph about the value? This is CO2 assimilation. Remember we described this. Yes.
Would it just be the amount of CO2 taken by the plant in low light conditions?
Okay, and so why do you think it is negative?
It's not getting the right amount of light, I'm not sure.
Okay, yeah, so it's not getting the right amount of light, right? It's in the light limited function. It's not getting the right amount of light. In terms of CO2, why is CO2 negative? What is that telling you?
So it's not in equilibrium.
So if a plant is not assimilating, so this is telling you how much CO2 is being assimilated by the plant. If the plant has a negative CO2 assimilation, what is that? What does that mean? What is the plant doing?
It's letting out more CO2.
Yes, it's letting out more CO2 than it's taking in. So that's what is known as that respiration rate. So the respiration rate, respiration as in mitochondrial respiration, that outputs CO2 is higher than the assimilation rate. So in this moment, what you actually have is CO2 being released from your plant. But can you tell me about the light compensation? Yes, well,
Would that just be like the minimum amount of light that is needed for the plant to be able to not be negative in the CO2 assimilation?
so yes, so the amount of light that is required for the plant to not be negative, or how? What else could it be?
It starts to create enough where. Sorry, what do you mean by that?
Yeah, so instead of saying for the plant needs to not be negative, can you state it in a different way?
It has its own production. It's
I mean, yes, you're right.
Yeah.
So what happens is basically at this moment is the amount of light at which the plant, the rate of photosynthesis, the rate of CO2 assimilation matches the rate of respiration. Okay? So you are at zero here. The amount of light at which, at least, yes, you're not really consuming CO2, but it's balanced. So what is happening here, and what does this mean for you? If you have a plant and you're measuring it, and you're obtaining this in this part, what can you tell me about that plant?
Photosynthesis is limited by the amount of light the plant's taking in.
Yeah, so basically that's what it is. So photosynthesis is limited by the amount of light, and the more light that you give, right? You increase this photo, the more light that you give, the more carbon fixation that is going on. Yes, Isabel.
If there's too much light, photosynthesis will stop, or the carbon fixation will stop. It'll not stop, but it'll even out.
Yes, so
I guess.
even out, yeah, so what happens to carbon fixation here?
So the amount of carbon coming in and the amount of carbon leaving is not changing. Okay, I guess I answered prematurely. I don't know what's happening.
So yeah, there's something about light, but not quite too much light. Yes.
The plant can't fixate carbon fast enough for the amount of light that it's taking in for photosynthesis to happen at a higher rate.
Yeah, so at this moment, what you have is light is no longer the limited factor. The limited factor is Rubisco or something else is fixing as fast as it can. So one of the things is that if it was too much light, probably you would see this going down because now you have the system shutting down, right? So maybe it's not too much light, but in this moment is carboxylation the limiting factor. So Rubisco is maxed out, something is maxed out, that is not light. You can increase light, but system cannot go any faster.
Um, so the shade plant is getting enough sun that it's producing. It's not able to keep up with the production of the stuff, and the sun plant is still able to go, but it's starting to flatten out a little bit to where it's getting to its max ability to produce.
Yeah, so what you can see here is adaptation from two different plants.
First
Right?
point.
You have is the light saturation points are going to be different. So when you have the shade plants, we talked about how some of these plants can be adapted. They get saturated and maximize their systems with little light, right? You increase light and CO2 assimilation increases that things are very efficient. They can be saturated with little light. A sun plant is adapted to get a lot of sun, and then basically the saturation is going to require more light. So I just wanted to mention something. This is right. So we've talked about light and somewhat about temperature. So the photosynthetic thermal optimum is basically that temperature at which photosynthesis, we talked about light, so now the temperature at which photosynthesis is going to be working at its best. And it depends on biochemical, genetical, and environmental components. The legends. Yes.
So basically, the difference between C3 and C4 plants is that obviously C4 plants are going to have like that waxy capable of sort of adaptation so they can handle a lot more heat. So they can also have a higher affinity for CO2 assimilation in comparison to C3 plants. And then obviously at a certain temperature, if it's too high, photosynthesis will stop because it's something that it's one of those limiting factors that we talked about previously. That's going to decrease with CO2 assimilation just because photosynthesis one to actually shut down.
Yeah, so it's showing you basically combining the things we've been talking about for the past 2 weeks, C3 and C4 plants, and how the leaf temperature and CO2 assimilation behave in these plants. So here in blue, you have the C3 plant in which it's doing very well, it's adapted to a range of temperatures, but pretty much above 30 degrees Celsius, it's probably doing a lot of photorespiration. Higher temperatures is already detrimental, and now you're wasting basically no more CO2 assimilation. You're actually doing photorespiration. You're releasing more carbon than you're assimilating. And C4 plants, here what you have is the plant actually benefits from higher temperatures. But as we know, anything that's related to enzymes, anything related to life, once you give it, well, in this case, enzymes, there's a temperature in which things are going to start going wrong. So, clearly, in terms of global warming, all of that, increasing temperatures, so we have C4 plants more adapted to that, but as with all systems, more CO2 is going to cause stomach to open less, that's going to cause less transpiration. The leaves will overheat, and then eventually that is, even when they are better suited for higher temperatures, there's a moment when the system eventually collapses. Okay? Good. Okay, so that's it. So, yeah, it's time. So I will have a great weekend and I will see you on Monday.
Alrighty.